Hello everyone and welcome to today's Daily Booster, which is number 7 in our series. To start us off today, we're going to have a look at the formula for force when we're talking about springs. So this comes up particularly when we're looking at experiments like Hooke's law. So the force in newtons is the spring constant times the extension. And the key thing to look for here is the extension units. So you can see there in meters. Make sure you have got meters in the question. Quite often they'll give it to you in centimeters. So before you can use this, convert to meters. For today's biology blast, we're going to be having a look at some of the defense mechanisms we have in our body against infection. So this is in B6, the second half of that topic, looking at diseases. So it will come up on biology paper 2. First thing we're going to look at are platelets. Now platelets are one of those 4 main components of your blood, and their whole purpose is to do with forming scabs. So what actually happens is if we cut ourselves, and we look at our first diagram here, so when you cut yourself, then obviously you start to bleed. Now, the first thing that's going to happen is we're then going to have these platelets passing through that area. So what we find is the platelets then change a protein in the blood called fibrinogen into fibrin. Now, fibrin itself is a fibrous protein, so it forms this network across the surface of the cut itself. Because we've got these fibers pretty much forming like a net, then the red blood cells get trapped in the fibers, and that is our blood clot. So when the red blood cells get trapped amongst the fibrin fibers, we have a blood clot. That's then going to harden to form a scab. And that means we're no longer exposing the inside of our body to the outside world. We've got obviously a covering over it there, which is going to reduce the risk of things like bacteria getting into your bloodstream. So make sure you know the stages there, that our platelets are all to do with blood clotting. And obviously if we have a lack of platelets, your blood doesn't clot well, which is problematic. Another component in our blood are the white blood cells. Now, white blood cells come in 2 types. We have phagocytes and lymphocytes. The phagocytes are the ones that carry out the process of phagocytosis, which we can describe as engulf and digest. Don't use other phrases. If you can't remember phagocytosis, then engulf and digest are the phrases we need to use. And the little picture down here just shows us what we mean. So the blue thing is our phagocyte. And what's going to happen is we've got these little red cylinders, which are our pathogens. And as our phagocyte comes in contact with them, it expands around them. It basically encapsulates them inside a little vesicle, so a little structure inside. And then it releases enzymes into that vesicle to break down the pathogen. We then just release the waste products, which are harmless. Option 2 are lymphocytes. These are the ones that produce the antibodies or the antitoxins, which are both protein molecules, which are complementary in shape to the thing they're going to attach to. So antibodies will attach onto antigens, and antigens are these protein spikes on the surface of the pathogen. So we have a lot of anti-words here that we need to go careful with. So our first one, the antigen, this is on the pathogen. So antigens are on the pathogens. The antibody, that is what's going to bind to the antigen. And then if we're talking about an antitoxin, they will bind to a toxin. So make sure you know those 3 different words. Antigen, the structure on the surface of the pathogen. Antibody is the protein that's going to bind to the antigen that our white blood cells make. And the antitoxin is the protein that binds to a toxin molecule. So what actually happens is when we detect an antigen, then our lymphocytes will produce these proteins called antibodies. And that can actually be one of those common questions. So they ask you, how are antibodies made? Then antigens are detected and the lymphocytes make the protein antibodies. Once they've been made, the next thing that happens is the antibodies bind to the antigens, which is obviously on the surface of that particular pathogen. And at that point, it makes it so that the pathogen can be ingested by a phagocyte. The process of that you will not need to know about unless you go on to do A-level biology, when we'll look at it in more detail. And the key thing about this is it's what we refer to as a specific form of defense. By that we mean each antibody is specific to one antigen. So for each antigen, you have to make a different antibody. Quick reminder of our daily formula before we move on to our chemistry. Force in newtons is the spring constant times the extension, remembering to make sure it's in meters. Today's chemistry check, we're going back to C4, again on paper 2 for our chemistry, to have a look at another group on the periodic table, the halogens, or group 7. First thing, you don't have to memorize the halogens. You don't have to memorize their names or their formulas, because in every chemistry exam, you are given a periodic table. So you can just read them. We have got fluorine, chlorine, bromine, iodine, and astatine. You'll find group 7, the second column in from the right-hand side. One thing we do need to remember, and the way that we generally throw away our marks on questions involving the halogens, is this bit here, that they're found as diatomic molecules. So that means when we come to write the formula, we have to put the little 2 in the bottom right. If you forget that, what's going to happen is any symbol equation you write is going to be worth 0 marks, because the formula will be incorrect and your balancing will be incorrect. So if you don't remember that the halogens go around as the little diatomic molecules, the little 2 in the bottom right, you're throwing away marks on any balanced symbol equation that involves them. So make sure you remember that. One of their favourite questions is asking you about what their colour and state at room temperature is. So fluorine and chlorine are both gases. Fluorine is pale yellow and chlorine is green. Bromine is the only liquid and it's orange in colour. Iodine and astatine are both solids, with iodine being a grey colour and astatine is a very dark grey, almost black colour. So make sure you do remember their states and their colours. In terms of the typical properties, these are non-metals, therefore they do have some typical properties of non-metals. They are brittle when they're solid, meaning they break quite easily, and they're poor electrical conductors. We also have some trends to remember as we go down group 7. Firstly, we've just seen they go from gas to liquid to solid. So as we go down, they move from a gas to a solid, which also tells us that their melting and boiling points increase. Hopefully that makes sense if they're going from a gas down to a solid as we go down that group. They get darker in colour and their density increases. So make sure you know those trends. In terms of their reactions, we do need to know of a couple of important ones. The first one is how they react with metals to produce a salt. So we've got our general word equation here. So remember, word equations, we have to write the full words. So metal plus the halogen makes a metal halide. Now this ending is important because if we mess up the endings on these names, you throw away the marks on word equations. So let's look at an example. If the metal is sodium and our halogen is chlorine, then it will be sodium plus chlorine, making sure that the halogen ends in INE when it's on its own. But when we write the salt name, write the name of the metal first, so sodium, and then because it's chlorine, we then change it to chloride. So any time the halogen is joined to a metal, the ending changes from INE to IDE. So watch out for those because it's one of the common ways people throw away the mark. If we look at the balanced symbol equation, then we start off with two of our metal atoms. They're going to react with one molecule of our halogen, and remember they go around as a pair, hence the X2, and that will form two molecules of our metal halide, MX. So let's look at our example at the bottom, again using sodium and chlorine. So we've got 2 sodium, 2 Na, plus Cl2, you must remember that little 2 in the bottom right, and that's going to make NaCl, and there'll be 2 of those. Again, this is one of those where if you really hate balancing, you can just remember 2 in front of the metal, 2 in front of the salt, and remember they're diatomic for the halogen. What we find is that when we're talking about the halogens, they will all have similar chemical properties because they all have 7 electrons in their outer shell. And this is a common question. It asks you why any elements in a particular group have got similar chemical properties, and the answer is always because they have the same number of electrons in their outer shell. In the case of group 7, that means they have 7 electrons, and they will therefore need to gain 1 electron in a reaction to make an ion with a single negative charge. And you can see the formula for that just at the bottom there. The reason that their reactivity is going to decrease as we go further down the group, the opposite to the alkali metals in group 1, is because where we need to gain an electron, as we go further down, we still have more shells, moving it further from the nucleus, meaning the force of attraction is weaker. So if we've got more shells, it's harder to gain that electron, therefore less reactive. Quick reminder of our daily formula. The force in newtons is the spring constant times the extension, remembering to make sure it is in meters. For today's physics prep, we're going to have a look at loudspeakers and microphones, which is the very end of our P4 topic, which will occur on paper 1 for those of you doing triple science, obviously. Now, when we think about microphones, first of all, we've got 2 types, and the one we're going to look at in more detail are the dynamic microphones. Now, the dynamic microphone, if we look at the little picture on the right, this is what it looks like. We've got a magnet, and around that magnet we've got a coil of wire. That coil of wire is attached onto this diaphragm at the front. So what happens is, as a sound wave, which is a pressure wave, comes in, it then hits the diaphragm at the front. As it does so, because it's a pressure wave, it pushes it inwards, and that means that coil moves down over the magnet. So what we find is every time we get that compression, the area of high pressure, it pushes the diaphragm in, and anytime we have a rarefaction, the area of low pressure, it pulls it back out. So we're moving the coil in and out over the magnet. As that happens, what happens is we are moving a coil of wire over a magnetic field, thereby cutting those field lines. And as a result of that, we're going to induce a potential difference across the wire. That is then going to travel down those wires, and it's an electrical signal that the microphone obviously produces. We'll get those differences in the electrical signal based on what's happening with the coil and its position over the magnet. But the key part is pressure wave, our sound wave, hits the diaphragm. That moves the coil over the magnet. As the coil moves over the magnet, we induce a potential difference across the ends of the wire, and that is our electrical signal. The second type of microphone we have is a carbon microphone, and these ones have carbon granules behind the diaphragm instead of that coil. So as that sound wave hits the diaphragm, what happens is it changes the resistance of the carbon, and therefore the current passing through those carbon granules changes because our resistance changes. The final device, our loudspeaker. If we look at the picture on the right again, so over here, we can see we've still got a magnet, we've got a coil of wire, and we've got a diaphragm. This is hopefully sounding very familiar to our dynamic microphone. And it is, because to all intents and purposes, it's kind of the opposite of a dynamic microphone. In this case, what we're doing is we're sending an electrical signal into the device itself. So what we have is a changing potential difference, which produces a changing current, and as a result of that, we produce a force on the coil of wire because it's in that magnetic field. So as a result of that, it moves the cone in and out, which then produces the sound wave. That's the basics of how our loudspeaker works, kind of the opposite of the microphone. Hopefully you found our little daily booster useful today, and don't forget to come back again tomorrow for our next one.